let's learn a little more about gene expression, how the instructions for being a particular kind of cell or organism, how those instructions are used to control the functioning of a cell, the functioning of an entire organism. And remember that all of the information that a cell or an organism needs to live, all the information is carried in the sequences of DNA in the chromosomes of that organism. And understanding gene expression helps us understand how all these genes, all these sequences, these segments of our chromosomes, how they are switched on and switched off to control the functioning of that cell or that organism. And understanding how these genes are switched on and off will help us to explain some of the great mysteries of life. How do multicellular organisms like us develop from a single fertilized egg and develop all of these really different shapes and sizes and functions in the cells of our body? And from moment to moment, how are cells able to change their functioning to respond to the different conditions in their environment, the different conditions inside their bodies and inside their cells? And answering those questions is all about answering and understanding how gene expression takes place in organisms. In the last video, we talked about operons, this model from bacteria that shows how groups of genes responsible for a particular function in the bacterial cell, how those genes are switched on and off, how, depending on the conditions in the environment, certain genes are switched on when they're needed, switched off when they're not needed, so that products are not produced needlessly. And we're going to extend that today to talk about some of the ways that more complex organisms like humans control the expression of our genes. So, a little review from last time. We talked about how these repressor proteins were able to block transcription, and so bacterial genes were switched off when certain molecules blocked the progress of RNA polymerase. So the enzyme that would transcribe the gene and start the process of making the proteins was prevented from doing so. There are also activator proteins that can bind to DNA and attract RNA polymerase and increase the transcription of a gene. Both of these types of molecules, which are virtually always proteins, are generally called transcription factors. So anything that binds to DNA and affects transcription is a transcription factor, a name in biology that kind of makes sense. And the places on DNA where those transcription factors bind are called enhancer sequences, and they go by different names. They talked about in the lac operans the operator region where the repressor proteins bind, but any place on DNA where a transcription factor binds would be an enhancer sequence. And they're typically pretty close to the gene, but not always, but typically they're pretty close to the gene, and in some way the binding of that transcription factor will help to switch on or switch off the gene. In eukaryotic organisms, the control of gene expression, like I said, is more complex. And if you think about the processes that we've just learned about with transcription and translation, at any point in that process, if we stop the process, if we switch it off, then we are controlling the expression of that gene. We're controlling the final product that would be made by switching on that gene. And at any step in the process, we could enhance that. That would sort of be turning up the volume. That can also affect the expression of that gene. So we looked at these models in bacteria, but we're going to talk about some different things in eukaryotic organisms. And this diagram has a whole lot going on, but what it's trying to point out is in the nucleus and in the cytoplasm, there are all of these places in the process of making a protein where the expression of the gene, the transcription, and then ultimately translation of that gene can be either inhibited, slowed down, or enhanced, sped up. And it starts with the chromosome itself and sort of packing or unpacking the DNA of a gene. And that chromatin modification that's at the top here, we know that DNA is wrapped around these proteins called histones. And when that DNA is all wrapped up and packaged up, it is not available, it's not accessible for RNA polymerase to get in there and transcribe a gene. So a way that cells can inhibit transcription from the get-go is to not unpack the sections of DNA where particular genes are. So when we have that condensed, compacted chromatin, that is switching off the genes in those areas of the chromosome. And it talks about histone acetylation here and DNA methylation. You don't need to know what those are all about chemically, but it's basically modifying the DNA or the proteins that are in these histone structures to package the DNA and to prevent it from being transcribed. At the level of transcription, we've looked at two cases there where we can block transcription and where we can enhance transcription by sort of clearing the path for RNA polymerase to transcribe the genes. Something we didn't talk about in bacteria, because it's not really important in bacteria, but it is in eukaryotic organisms like us, and that's RNA processing. The messenger RNA that is transcribed generally has a number of introns in the messenger RNA that are not part of the instructions for building a protein. Those introns have to be chopped out by enzymes in the nucleus to create a final messenger RNA that's sort of ready for the ribosome. And so we could block the process there by stopping RNA processing. Once the RNA gets out to the cytoplasm, we can block the expression of the gene by breaking down that messenger RNA quickly or not so quickly. If the messenger RNA stays around in the cytoplasm longer, that gene can be translated into more and more proteins the longer the messenger RNA stays around. So we can degrade or break down the messenger RNA quickly to slow down or limit the expression of the gene. And then after the protein is made, we can also enhance or inhibit the expression of a gene by messing with the process of finishing the protein, packaging it, and sending it to where it needs to go. All of these steps in making a protein are areas where the cell can potentially intervene and enhance the process or slow it down. Where would it make the most sense to control the expression of genes most often? Like in terms of efficiency, it would make the most sense to control the expression of genes up here at the top of the process. Because once you transcribe the gene, once you process the messenger RNA, once you've built the chain of amino acids, a whole bunch of energy and cell resources have gone into each step in that process. So it would make sense that for most of the control of gene expression, we do it in these first steps. And that's, in fact, what happens. But it's possible to control in all those latter steps as well. Okay, let's look at an example of controlling gene expression in a moment-to-moment situation. And we're going to look at how a hormone in a eukaryotic organism like us can affect gene expression to help maintain homeostasis. Homeostasis is this huge concept that we talked about very early in the year, so you may need to refresh your memory about that, but it's basically the idea that living organisms work their rear ends off to try to maintain conditions inside their bodies and inside their cells that are optimal for the cells to function. And maintaining that stable environment, those stable conditions, and all the functions that go towards maintaining those stable conditions are under this blanket term, homeostasis. And so a lot of controlling gene expression is directly related to homeostasis, maintaining the stable conditions in our bodies that are the best conditions for our cells to thrive. So let's see how this works with a hormone. And I have chosen testosterone, but it could be any of the steroid hormones, progesterone, estrogen. There are a whole group of hormones in this category that are lipids. And this makes it a simpler example because the hormone can pass through the lipid bilayer. It's soluble in the plasma membrane. So when a hormone like testosterone reaches the surface of a cell, it's possible for it to pass right through that cell membrane and enter the cell to start to affect the functioning of the cell. So our hormone has gotten past the cell membrane, and inside the cell there is a receptor protein for testosterone. And that receptor has a binding site that's very specific for this hormone. So testosterone gets into the cell, it bumps into this protein, and the hormone and the protein bind to one another and become a hormone receptor complex. That hormone receptor complex has the ability to get into the nucleus, and it has become a transcription factor. Because when the hormone is bound to this receptor protein, this receptor protein has changed its shape and now can bind to certain sequences on DNA. It has become a transcription factor. When the receptor protein did not have the hormone bound to it, it was not a transcription factor. It was just a receptor floating around the cytoplasm. Now, once that hormone receptor complex binds to the DNA, it can do what we saw the transcription factors do in the bacterial operons that we looked at. It can either switch on or switch off a gene. And in this case, it's showing the example that the hormone receptor complex, this transcription factor, is an enhancer. It activates a gene and it stimulates the production of a messenger RNA, which can go out into the cytoplasm and affect the production of a protein. And that protein would affect some change in the cell. That protein would be the consequence of that hormone reaching the cell. So that hormone may have been released because there is some imbalance, and the cells need to change their functioning to correct that balance. And making this messenger RNA, making the protein that comes from that messenger RNA, would be the cell's response to that signal and would hopefully correct whatever imbalance may have existed. So this slide is, again, summarizing some of the big ideas that we've talked about. Genes are regulated lots of different ways by these transcription factors that can bind to DNA. The places on the DNA where these transcription factors bind are called enhancer sequences, and they can be things like the operators in the like operon, or they can be enhancer regions, activator regions. They go by different names. In any case, it's a place where a protein can bind to DNA. And then that binding affects the transcription of genes that are in that area of the DNA. It can either open up some tightly packed chromatin so that the genes that were wrapped up there are now exposed and could be transcribed into messenger RNAs. Those enhancer sequences might attract RNA polymerases. RNA polymerases might gather next to those enhancer sequences when they bind to the DNA and bind then to the promoter regions and start transcribing genes. And the enhancer sequence may bind to the DNA and block messenger RNA from transcribing genes. So general ways that these transcription factors can control the expression of genes.